Okay, hey, so my question was, tell us about the 3 types of mutations that occur spontaneously during replication. Are these mutations passed on to offspring? Why or why not? So the DNA alterations are occasionally beneficial because DNA-based sequencing changes or mutations provide genetic variability, and so the 3 common types are spontaneous mispairing of bases via transient formation tautomers, slippage during replication, and spontaneous damage to individual bases. So the most common form of spontaneous replication error is through the mispairing of bases via transient formation of tautomers. Tautomers are rare alternate resonance structures of nitrogenous bases. When a base is in its rare tautomeric form, it can pair in a non-standard way with other bases in a process known as tautomeric shift. The result is a new daughter strand that actually does carry the incorrect base in that position, and it will continue to be transmitted to the complementary strands throughout eventual replication. So I don't have like a whiteboard or anything, but this picture made a lot of sense. And so here it shows the regular base pairing, and so and then down here it shows the rare tautomer. So it's whenever it forms this extra bond right here, and that's not what it normally does. And so and that would be carried out throughout replication. And so then the next one is slippage during replication. And it involves regions of chromosomes that contain highly repetitive DNA. An example of this involves trinucleotide repeats, which are especially susceptible to strand slippage, which is when the DNA polymerase replicates a short stretch of DNA twice. If DNA polymerase detaches from a newly replicated region of repetitive DNA, then the repeats may form a transient hairpin, exposing a stretch of the template that has already been replicated. The result is that when the DNA polymerase reattaches and resumes replication, the same region is replicated. The specifics of the disorders caused vary, but the accumulation of extra trinucleotide repeats in specific genes can result in abnormal proteins that lead to cellular defects or the reduction of levels of expression in the associated genes. And so once again, this was just a really good picture that shows the whole process. So up here you have the DNA segment and all the trinucleotide repeats, and then the strand separates, and then we have the replication end. So down here, this is the hairpin loop whenever it does the accumulation of extra replications, or no, extra trinucleotide repeats, and so then that ends up changing the whole sequence and what is produced at the end. And so then the final type is the spontaneous damage to individual bases. It involves chemical modification of the bases. The most common is depurination and deamination, which are spontaneous hydrolysis reactions caused by random DNA and water molecules around it. Depurination refers to the loss of a purine base, which would be adenine or guanine, by spontaneous hydrolysis of the glycosidic bonds that link it to deoxyribose. Deamination is the removal of a base's amino group, which would be the NH2 or 3. Yeah, the NH3. Which can involve cytosine, adenine, or guanine, changes in base pairing properties of the affected base. If a DNA strand with missing purines or deaminated bases is not repaired, an erroneous base sequence may be propagated when the strands serve as a template for the next round of DNA replication, but fortunately cells use several repair processes to dramatically reduce the frequency with which permanent defects in DNA are passed along during replication. And so here, just very simply, this shows, so here you have your guanine and deamination, so it's just literally removing that group. So then over here on this other side, you have the result, but it's missing this guanine base. And then for deamination, it takes this little amine group off of the cytosine. And so over here, you have that has been turned into uracil, and it is missing that amino group. So those are the 3 types of mutations that occur spontaneously. And yeah, so thanks.